Okay, so we have taken a look at the dermis. We've seen that thin skin has 4 layers, thick skin has 5 layers, including that stratum lucidum. We are able to compare thick skin versus thin skin, and you're going to be able to tell me which is which. We've also looked at darkly pigmented skin, and you can tell me which one is dark and which one is light skin. So melanin is going to contribute to skin color. Melanin is one of the primary factors in our skin color. And melanin comes in different shades. There's not just one kind of melanin. There's some melanins that are very tan in color, some that get progressively darker. So it gets to more of a shade of brown, maybe a yellow tint to it. It gets darker and darker brown until we have some melanins that are very, very black. And some have kind of a red color to the skin, or to the melanin. So we have these different shades that are going to contribute to skin color, which is how, depending on your heredity, you have a different skin color maybe than somebody that you're sitting next to. There are other factors that contribute to skin color. There's another pigment, carotene. What color is carotene? Orange, yeah. So it really is orange. It is the pigment that makes carrots orange. And you can actually get too much of this, and your skin can actually begin to collect that pigment, and you can kind of become orangey color. Ask me how I know. I know because my first baby, when I started feeding her baby food, loved the oranges and the yellows, right? The carrots are great, the squash is great. Peas, nah. Peas always ended up out here. And the green beans, no way. So I just, sing. So I thought, well, I gotta eat carrots too. And when I took her in for one of her first baby checks, the pediatrician was like, do you feed her a lot of carrots? And he says, yeah, because she's turning a little orange. And I just thought she was this beautiful, natural, like, she has a natural skin. No, you can actually get too much carotene. He says, you really need to feed her some green things. So that does contribute to skin color. Also, hemoglobin is another pigment. Hemoglobin is red, and that is found in our blood. And so our blood actually is coming to the surface, and that is going to impact our skin color as well. We're going to learn more about hemoglobin in 2102, ANP2, when we talk about both blood and then about the respiratory system, because hemoglobin is the pigment that carries oxygen as well as carbon dioxide. So more to come about that. There are some other skin color phenomenon that are not homogeneous. So it doesn't cover the entire skin, but we have little parts of the skin that can change color. And so you've heard of moles. We probably all have at least a mole, or some of us have lots. And so a mole is called a nevus, which is a little spot of dark color. It is typically perfectly benign. It's just where more pigment is being expressed in that area. But if it changes in appearance or in size or in elevation, how thick it is, that could be a sign of a malignancy, and that would need to be checked out as soon as possible. Your doctor would get you in right away if you had a concern about that. Freckles, some people have more freckles than other people. And again, this is just more spots, more localized area where there's more melanocyte activity. And then as you age, you may see more and more hemangiomas. That oma is a little scary because it's like, oh, that sounds like cancer. But this is perfectly benign, and but it is a little tumor, and it's in the blood vessel. And so that's going to be a red spot. And as we age, we just get more of those and have more red spots, but they are perfectly benign. So these are all colors of skin that you may or may not have right here. Sticking with epidermis, let's look at this one. And this doesn't show well, but in the epidermis, we have ridges, epidermal ridges. And those epidermal ridges are going to be more pronounced, particularly in thick skin, and they are going to contribute to your fingerprints and your toe prints, right? So epidermal ridges are going to provide what function? What do they do for us? I mean, why do we have fingerprints? It's not for identification. Grip. Yeah, it's friction. Epidermal ridges are going to provide friction Does that make sense from an adaptive perspective? Yeah, I mean, we need to be able to use our hands. That's why we stood up, right? So rather than walk around on our knuckles, as some of our ancestors did, we stood up, we became truly bipedal. And now look at the freedom we have. So I can walk with my legs, but look at all of the things I can do that my dog cannot. Thank God. Okay. First of all, we have thumbs, opposable thumbs, and we have fingerprints, which allow us to increase the utility of our hands. So our ancestors may have been swinging from trees, and they've got to have that grip. So how did this first start to evolve? Well, the ones that grabbed the branch and were able to hang on survived. Those that went, oh, shoot, may not have survived, either because they fell or because the leopard was at the bottom of the tree waiting, right? So those epidermal ridges persist and they are helpful to us. And conveniently, they are unique to us. So we can use them for identification purposes. All right, let's move now to deep to the epidermis and look at the dermis. Our dermis has two layers. These are a little more subtle than the epidermal layers under the microscope, but you can see them, and we will in just a moment. So the top, let's see, the apical, or I could also say the more superficial 20% is called the papillary layer. And the bottom, I'm going to say superficial because this isn't epidermal tissue, so it's not really apical and basal. So I'm going to say superficial and deep. The deeper 80% is called the reticular layer, which is unfortunate because it is not reticular tissue. So the papillary layer is made up of areolar tissue. And the reticular layer is made up of dense, irregular connective tissue. Looking at this image, we have the epidermis peeled away a little bit. And that allows us to see the top superficial most part of the dermis. And see how it's bumpy? Each bump is called a papilla, but we want to be specific. So in anatomy, bumps are papillae. Technically, it means nipple. Plural is papillae with an E on the end. But we want to be specific because there are so many papilla that we will be identifying throughout A&P 1 and 2. And so we are going to call these dermal papillae. And these papillae are going to illustrate an anatomical property we are going to see over and over and over again. We've already talked about it when we looked at some of their tissue types, and that is surface area. Does having bumps increase or decrease our surface area? Definitely increases it, yeah. So let's look at a road trip. I'm going to drive from point A to point B, and my first option is to take the road that goes straight there like this. My second option is to go through the mountains. Which one is going to put more miles on the car? 1 or 2? Definitely 2. Yeah, but it's going to be more scenic, right? So more surface area you can see here. So why do we need more surface area? Because it's going to allow more surface for the epidermis to stick to the dermis. So it's going to increase the ability of that epidermis to stick to the dermis. And so we'll have this basement membrane covering all surfaces. And there's something else happening here. Remember, epidermis is what kind of tissue? Stratified squamous epithelial tissue. Epithelial tissue, vascular or avascular? Avascular, no blood supply, which means this tissue must get its nutrients and other materials from blood supply in the underlying tissue, which means our dermis. And these papilla project up into the epidermis so that these blood vessels, which are in the papilla, are able to penetrate the epidermis, even though the epidermis doesn't have its own, which means that more of these cells are going to receive the nutrients, the respiratory gases, and the other materials that we'll learn more about in A&P 2. Notice that also some of these papilla have sensory receptors in there. We'll talk about more in a little bit. And so those sensory receptors are a little bit closer to the surface of your skin also. So those papilla have multiple functions or repercussions. They also make up the papillary layer of the dermis. So the papillary layer probably comes down to here. So it goes a little below the level of the papilla. And then everything below that is the reticular layer, which is not reticular tissue, it's dense, irregular connective tissue. When you look at the slides to look at the dermis, start on that low power. Start using that 4 times objective and see if you can see the difference between the papillary layer, which will be a little bit more smooth pink, and the reticular layer, which is going to be more swirly pink, a little more white space in there. And just see if you can see the difference. On a microscope, if I'm going to ask you which layer of the dermis is this, I'm going to make sure I put the pointer way up in the papilla or way down here. I'm not going to be subtle and put it really close to the line between the two. As you zoom in, it gets a little bit harder to tell the difference. Looking at the dermis, we see a lot of stuff going on in here. So let's look at some of the structures that are contained in the dermis. Some of those structures are sensory receptors. Remember, the epidermis had the Merkel cell, which McKinley calls the tactile cell, that is connected to a sensory neuron that carries that sensory information to the spinal cord and then from the spinal cord up to the brain. We have other sensory receptors in the dermis that are also connected to or communicate with sensory neurons. Up here, these little ones are Meissner's corpuscles. If you say corpuscles, you'll remember to put the C in there. Meissner's corpuscles, and then very deep down in the dermis, we have more tactile receptors, McKinley. These are Pacinian corpuscles. Pacinian corpuscles very deep and are able to detect deep pressure. Meissner's corpuscles can detect much lighter pressure in some vibration. There's another kind of sensory receptor in there, and it's actually part of the sensory nerve, and it's called free nerve ending. Typically, the free nerve endings are detecting some chemicals that are present in that local area, and often those are associated with painful stimuli. Ow. That's more likely to be a free nerve ending than a corpuscle. Our author has really simplified the tactile receptors, puts them all in a group. They're all tactile receptors or tactile cells. But for lab, let's just put our corpuscular language in there. So very much a sensory organ, our skin. Does that make sense? Yeah, it's interacting with our environment. We need to know about our environment. I need to know if I'm touching something hot. I need to know if I'm touching something, right? Pressure. I need to know if somebody just smashed a hammer on my hand. So that communication is going to be very important in my interaction with the environment. Also in the dermis, we have hairs and hair follicles. So let's look at those. Let me just sketch a hunk of skin. So here's my dermal papillae. Up here's my epidermis. So this is all the epidermis. Down here's dermis. And now I'm going to take my thumb and I'm going to push it down into the skin. And when I do that, these layers invaginate like this. and the papilla goes up inside there. So this hole, this container, is what we call the hair follicle. The hair follicle can contain a hair. But the follicle itself is not a hair. It is a container for the hair. The hair itself has the shaft. The shaft is everything superficial to the skin. And then the root of the hair is everything below the surface of the skin. But I want you to focus on this area right here where that hair is enlarged at the base of the follicle. This is what's called the hair bulb. The hair has epithelial cells, and the living cells are here in what's called the matrix of the hair bulb. So the hair bulb contains living hair cells. And as those cells divide, the older cells are moved superficially. So that by the time the cells emerge from the surface of the skin, those are dead cells. Here's our living cells. But if they're epithelial cells, vascular, avascular. Avascular, right? So where's the blood supply come from? Well, once again, we have a papilla that has blood supply. But this is not a dermal papilla because it is not in the dermis. So we are going to call this the hair papilla. If I were to ask you this on a test or this, and you say papilla, that is good for half a point. But if you say dermal papilla or hair papilla, you've got it. All right, I have a question for you. Given what you now know about hair, what's the best shampoo we could possibly use to give our living hair cells the nutrients they need? Shampoo goes up here, right? Is this living or dead? Dead. Are nutrients going to be absorbed by those dead cells? Probably not. Probably what we want to put up here are materials that are going to take the little cells that have flayed out to the side and smooth them in back together so we don't have the frizzies. But these are the cells that need the nutrients. So how am I going to get nutrients to these cells? Through the blood. Through the blood. And how do I get nutrients in the blood? I consume them, right? Anything I get into my blood is going to go everywhere the blood goes, including here. So the vitamins that I consume, the nutrients that I consume can impact those living cells. That's how I improve the health of my hair or the health of those cells. All those 14 or 16 vitamins and minerals, unless they coat the hair, it's unlikely that they're getting all the way down in here, right? But shampoo companies will never tell you that. It also means that anything else you put in your blood, medications, toxins, are also going to be exposing those cells to those materials, right? So it can have an adverse effect. How about something like chemotherapy that causes, in many cases, hair loss? Yeah, chemotherapy is chemicals injected into the blood. And that chemotherapy is meant to kill cells that divide rapidly, i.e. tumor cells. Tumor cells divide and divide and divide and divide and divide rapidly. And that's why cancer can be so dangerous, because those cells divide at the cost of all other living tissue. So we want to kill rapidly dividing cells. Well, you know what other kinds of cells divide rapidly that aren't unhealthy? Epithelial tissue, like our hair cells, like the lining of your gut. Because what's another side effect of chemotherapy that impacts your gut? Yeah, nausea. You can't taste things, your taste buds get all wacko. And the hair falls out. That's all the impact of chemotherapy on these epithelial cells. So including hair loss. Makes sense? I know, is it coming together a little bit? Yeah. You know what else impacts hair growth? Hormones. Hormones definitely have a huge impact on the health of your hair. And let's talk about a certain genetic condition which can impact the presence of hair and the shedding of hair. And that is a gene that contributes to something we call male pattern baldness. Heard of this? Yeah, this is where usually in men, almost always in men, they have a very defined pattern of hair loss. There's different kinds of baldness. But in this one, the man is losing the hair usually on the top of his head, not so much on the side. He's able to grow a full beard. He's able to have body hair. But the hair on the top of the head is lost. And what's happening is this. In male pattern baldness, first of all, it's a delayed onset gene. So if he has the gene, then it could turn on later in life. It might turn on before puberty, but it might turn on when he's 40 something. And then he's in the shower going, what is happening? Right? And all of a sudden started losing my hair. That gene may have just turned on. That gene causes the hair follicle to become sensitive to testosterone. And when exposed to testosterone, the follicle lets go of the hair. It sheds it before the hair grows to the surface of the skin. So the hair is in there, it's growing, it just doesn't stay before it reaches the surface. It sheds, as all hair does. So hair loss before it reaches the surface. This is an X-linked gene. It's on the sex chromosome, but it's the X chromosome. Didn't we say that baldness, does baldness come from your mom or your dad? Have you heard that it comes from your mom? Yeah, X, but doesn't dad have an X? Okay, here's what's happening. So women have two X's, men have an X and a Y. If there is a male pattern baldness gene, we'll just put a little B on there to represent that gene. If a woman has that X with the little B, then she may have another X that's dominant. And so this gene isn't expressed because the dominant gene is going to be expressed. But if a man gets that X, maybe from mom, then there's no other X with the dominant gene to mask that. If he gets the gene, he will express it. That's true for any X-linked gene. It's why color blindness is more common in men, because it's an X-linked gene. Some forms of hemophilia are X-linked genes. They're more expressed more often in men, because if the man has it, he's going to express it. If the woman has it, she's got to be homozygous. She has to have both X's have this to express the gene. So if he's got the gene, he's going to express it. Now, is that an impossible gene combination? No, there's probably a lot of women who have that. But we don't see a lot of women who have this pattern baldness. That's why it's called male pattern baldness. Why not? There's women out there who have this. Why don't they have pattern baldness? Because
testosterone is the weak that breaks it.
That's right, because how much testosterone is she making compared to him? A lot less, right? And so the follicles are going to be sensitive to testosterone. If she were to do a testosterone shampoo, if she were to take testosterone, I just learned that there's testosterone pellets that they'll implant in women for some conditions, and sometimes their hair starts to fall out. Oh, maybe she has that gene. So hormones and nutrition are going to affect the growth of the hair. We're going to mention this again if you're in my class for A&P2. We'll talk about this again in the context of genetics at the very end of A&P2. What's hair good for? Say again? Temperature regulation. Yeah, it keeps us warm. It's not so good for us as it is for our furry animal friends, right? And some animals have hair that has that's hollow, which is even more insulative. Is that a word? Has more insulation properties. So otters, polar bears have those hollow hairs, which also allows them to float better. What was the other one? What is it? Protection. A little bit, yeah. A little bit if you've ever been bashed in the head. Or my son, or from mosquito, they like this. Say that you have from my son, it protects the sun so you don't burn. And you learn that if you don't have hair, you know, if you are bald or if you have shaved your head. You bet. But we have thumbs and we can make hats. So we're lucky. I would add aesthetics. That's probably not in the book, but does that have a function? All right, remember, all of our anatomy and physiology has adapted for our survival of the individual, but also survival of the species, which means you have to reproduce, which means you have to attract a mate. I don't know. Maybe those otters are out there saying, oh, that otter has some really cute little tufts of hair. Ooh, ooh, ooh. Right? I don't know. Oh, I'll tell you one more thing and we'll take a break. This is not on the test, but these are things that we need to consider. So it's been a while since I've seen any advertisement, and I don't know if the product minoxidil still exists. Anybody know? For a while, it was the first hair growth product before Propecia. Minoxidil came out, and they made it as a shampoo, or I think, yeah, it was a cream. And on the commercials, because they advertised it to make this was going to be the money maker, they had the disclaimer, as all medications do, women should not handle this cream if they are pregnant or nursing or expect to become pregnant. So what, you have a hairy baby? What's going on here? Well, here's the backstory. And I don't know all the details, but minoxidil was originally in development as a blood pressure drug. I've worked in biotech. I know how this works. I was like pair of hands number 4. I don't know. Pick up the rat, put it in. And so I'm sure the lab techs were like taking blood pressure measurements, and you report to the scientist and say, you know, blood pressure in the control rats versus experimental rats is not changing. But men's rats are hairy. And somebody went, bing, I have an idea. Market as a hair growth drug. And they did. And the mechanism of this drug, turns out it blocks testosterone receptors so that the follicles are no longer sensitive to testosterone and the hair doesn't shed. Yes. Where else do we have testosterone receptors? Well, lots of places. What else is testosterone involved in? Well, among other things, sex drive. In the developing fetus, or embryo, I guess I should say, the developing embryo, the male and female embryo is pretty much the same. But what changes is if it has a Y chromosome, it begins to secrete testosterone, which binds to the testosterone receptors, and that little embryo becomes a fetus that will develop a penis. But this little embryo that doesn't have the Y chromosome doesn't make as much testosterone, and so doesn't develop a penis and retains its clitoris. Huh. So there's something. After puberty, testosterone contributes to the secondary male characteristic. Deep voice, hairy body. Okay. So if this drug gets into the blood, as it does, it's transcutaneous absorption into the blood, it's going to go everywhere that blood goes, which is pretty much everywhere, including the tissues that have testosterone receptors. And if this drug blocks testosterone receptors, it could have an impact on those tissues. And that's why in the fine print, one of the side effects of the spidoxydil cream, potential drop in libido, sexual. Now we've got to decide hair, sex drive, hair, sex drive. Okay, you see the problem there? All right, well, how about the woman who's pregnant? Puts this on her hands, it's absorbed into her blood, and it blocks the testosterone receptors in that developing embryotic piece. You can't say, like a little girl baby. You know, doesn't work that way. There are potentially horrific birth defects. from that medication. And I don't know for a fact, but I wonder if that's why we don't hear about this medication anymore. But it was a hair growth, it was billed as hair growth medication. So we can develop these drugs that do all these amazing things, but you've got to consider the unintended consequences. There's not a magic bullet that'll go right to the receptors you want to target and only there. And a lot of scientific discovery and research is on delivery mechanisms. Okay, I spent a lot of time doing that in biotech world. How do we get this drug to go here? And where's the overflow go? And what effect is that going to have? Things to consider. Let's take a little break.